Hello students, welcome to class 12 biology. This is part 3 of biozoology 5th lesson, that is molecular genetics. Last class we have learnt about the structure of the DNA. Now we shall go in detail of replication of DNA. There are few methods by which the DNA could replicate that is being proposed by scientists. They are conservative, semi-conservative and dispersive methods. So you can just follow the diagram, and we have learned about replication of DNA semi-conservative model. So that is one of the most accepted model. Semi-conservative model of DNA replication is mostly accepted. Here we have the two colors of DNA, that is blue and yellow. So this yellow color denotes that it is a daughter DNA that is being produced from the parent. That is a replica, which is a similar structure of the parent one. Okay, so there is no difference between the parent and then the offspring. According to conservative model, it is said that through some means it is able to produce the copy. So the scientists were not able to say by what is the method by which it is producing the daughter DNA. But semi-conservative model, it clearly explains how this DNA unwinds and it is making its copy so that it could produce the daughter DNA. See, semi-conservative model, half of the parent DNA was conserved because it is preserved as such, and the template copy is being taken from that parent. So that is semi-conservative model. Dispersive model, here the DNA is getting cut into pieces and it is forming the complementary strand and they stick together to form the daughter DNA. It is also not accepted. DNA only copied itself for short chunks. Okay, how these short chunks are being produced? By being cut. Okay, so short chunks at the time producing new DNA strand that altered the parent and daughter DNA. Okay, so only the semi-conservative model is most accepted. Here we have the experimental proof saying that what is being happening during the replication of DNA. Okay, for that we are using the material called N15 and N14. What is N15 and 14? They are radioactive element. Once we use radioactive element in our experiment, it is easy to find the presence of the molecule. For example, if you have the DNA that is being treated with the nitrogen 14, so then you put all the DNA together and which is being treated with N14, you put with along the other DNAs. Okay, so now because it is radioactively labeled, we could easily isolate them. Do you follow? This radioactive element helps in the isolation, segregation, finding. Okay, so here there is an experiment conducted to find how this DNA is getting replicated. Okay, so that is proposed by Messelson Satal, that is using nitrogen 15 and nitrogen 14. First, he has taken nitrogen 15. Bacteria were grown in the medium where there is N15. So, if there is N15 being present, all the bacteria will possess N15. Then, it is one E. coli is being taken. E. coli is Escherichia coli bacteria. That is taken and it is put onto the another medium. That medium contains only N14. Okay, so this bacteria will undergo replication. Once it is undergoing replication, it is producing the daughter cell. Till that, is it clear? So N15 one medium, bacteria is grown. So bacteria has incorporated N15 into it. Then this bacteria is taken and it is put into another medium where N14 is present. So this N14 should get incorporated. N14 also should get incorporated with bacterial cell. Okay, so this bacterial cell will undergo replication or it will produce daughter cell as usual using the nutrient in the medium. Follow? So then this DNA is being checked for the presence of N15 and N14. If you find one strand having N15, another strand having N14, only semi-conservative model is accepted. If you find both the strands are having N15 and both the strands are having N14, then the conservative model is accepted. If you could find both N14 and N15 in the same strand, then this dispersive model is accepted. Do you follow the purpose of this experiment? Okay. So the how it is being done, that is what is said here. Then after the result is being drawn, it is said that the semi-conservative model, one DNA strand is the parent one, another DNA strand is the daughter's one. Okay, so with that they conclude that the semi-conservative method of DNA replication is found. Then we have comparison of replication with prokaryotes and eukaryotes. It occurs, prokaryote shows it occurs inside cytoplasm. Eukaryotes, it occurs inside nucleus. Single origin of replication. What does it mean? Origin of replication. The place from where the replication starts. Origin. What does it mean? To start. The place from where the replication starts. So only one place is there for the start of replication, but in the case of eukaryotes, in any place there are numerous places from where the replication could start. DNA polymerase III carries out both initiation and elongation process. Initiation to start, elongation to proceed. Okay, and final process is called as termination. You should know all these three terms: initiation, elongation, termination. Initiation to start with, elongation is the process, and termination is the final stage. DNA polymerase, it ends with ASE. As I told you, ASE, if the word ends with, it is an enzyme. Very good. Okay. So then that is being done by DNA polymerase III. Then here in eukaryotes, initiation is carried out by DNA polymerase alpha and elongation by delta and epsilon. Okay, so eukaryotes require different enzymes for initiation and elongation. DNA repair and gap filling are done by DNA polymerase 1, and here it is by DNA polymerase beta. RNA primer is removed by DNA polymerase 1. RNA primer is removed by DNA polymerase beta. Yes. So here RNA primer that we will learn in detail. Further we will learn about RNA primer. So RNA primer helps in the replication process. That is why it is discussed here. Then Okazaki fragment. Do you find the 6th point? Okazaki fragment. So it is a very small fragments that is formed as a daughter nucleus. See, once from the parent DNA, daughter DNA has to form. Once this daughter DNA is getting formed, it is not a full strand is getting formed. Bits and bits is getting formed. Those fragments are called as, what are fragments? Small bits are called as the fragments. So small bits are called as the Okazaki fragment. Replication is very rapid in the case of prokaryotes and it is comparatively slow in the case of eukaryote. DNA gyrase is needed and DNA gyrase is not needed. What is DNA gyrase? It ends with ASE. So it is an enzyme which helps in the process of replication. Understood? Okay. So here you can see steps in replication. So prokaryotic cell, recognition of origin of replication. It is present in DNA A protein and unwinding of DNA by helicase enzyme and stabilization of unwound templates. So it has to stabilize. Once it is getting unwound, okay, it has to just um, get into single strand from the double strand. Okay, single strand DNA binding protein SSB is present. Okay, so next, synthesis of RNA primer. RNA primer should be synthesized, that is being taken care by primase. What is primase now? Primase is an enzyme. Why? Because, yes, good. It ends with ASE. Very good. Please do remember, because you should not think that you do not know the meaning for certain words. When it ends with ASE, it is enzyme. That's all. Okay. Next, synthesis of DNA. So, leading strand and lagging strand, two strands should be produced. That is taken care by DNA polymerase 3 enzyme in the case of prokaryote. Removal of RNA primer. So, RNA primer, after a replication process, it should be removed. That is done by DNA polymerase 1, or it is exonuclease. 5-2 3-end exonuclease. Now we will see for eukaryotic cell. So origin of replication is unknown. Why? Because there are more than one origin of replication. So you do not know which is the origin of replication. Next, unwinding of DNA double helix is taken care by the enzyme called helicase. Good. Then second one, stabilization of unwound template strand, that is taken care by SSB, that is single strand binding protein. Synthesis of RNA primer is taken care by primase. Leading strand is by DNA polymerase delta and lagging strand by DNA polymerase alpha. Then removal of RNA primer, how it is done, we do not know. And replacement of RNA with DNA also is not known. And joining of Okazaki fragment is done by ligase, as we saw in the case of prokaryote, and requires ATP. So it requires the presence of energy. Then removal of positive supercoils. What is supercoil? It is highly coiled region of DNA. I repeat, supercoils of DNA is a highly coiled region in DNA that has to unwind so that the DNA can proceed with the replication process. That is done by topoisomerase enzyme. Next, so it is another name for DNA gyrase, which we have seen in the last tabular column. Yes. So DNA replication, stepwise it is explained here. We'll start with the first step, DNA replication initiation process. So initiation by helicase. So first the double strand should get into the single strand. Okay. So to start this process, it is forming the replication fork. Because of the action of the enzyme, it shows fork-like structure on DNA. Okay, so that is called as replication fork. Multiple replication forks along the DNA molecule creates replication bubbles. Do you follow? Replication bubbles, how it is getting formed. First, the origin of replication, helicase enzyme is it is getting into the single DNA strand. Double DNA strand is becoming single. Okay, so it is forming the bulged region in the middle. That is called as replication bubbles. Next, step 2, RNA primase. It is an enzyme. Adds a short segment of RNA called RNA primer. Okay, so it is what is RNA primer? It is a small segment of RNA. It is attached with the DNA during the process of replication. It gives DNA bases something to attach when they are laid down. Okay, so once they are coming, they need support that is given by this RNA primer. Okay. So this process, helicase is doing its process. Do you follow? So replication arrow is given. So first helicase is doing its work. At the back, RNA primer is going. At the back, DNA polymerase is doing its work. Okay. So it is just moving to form another strand. Okay. Next step 3. The strands of DNA are being copied in opposite direction. That is called as the leading strand gets replicated in one continuous piece. Leading strand is a replication in one continuous piece. Then lagging strand is replicated discontinuously into small pieces called Okazaki fragment. Once it is getting into small pieces, Okazaki fragment. Okay, so that is about the replication process. Now we have this DNA replication along with this transcription and translation. Follow? So from DNA, mRNA should be formed. Then mRNA, tRNA, rRNA together forms this protein. That process is called as DNA to RNA. The process is called as transcription. From RNA to protein, it is called as translation. Okay, so then we have particulars, transcriptions and reverse transcriptions. So this occurs in both prokaryotes and eukaryotes show the transcription. Reverse transcription is seen only in the virus. What is transcription? It is the formation of RNA from DNA. If RNA is giving rise to DNA, then it is called as reverse transcription. Follow? Transcription, DNA to RNA. If DNA is formed from RNA, it is called as reverse transcription, that is seen only in viruses and some of the viruses. Molecules synthesized during transcription are RNA from DNA, reverse transcription DNA from RNA that we have discussed. Enzyme involved is transcriptase, here reverse transcriptase. Template use DNA, reverse transcription RNA. So if you could understand, you need not by heart, you can write on your own. Okay, so now we shall go in detail of transcriptional units. So transcriptional units sequence of nucleotide in DNA that codes for single RNA molecule along with sequence necessary for transcription. The stretch of DNA transcribed into the RNA molecule is called transcription unit that codes at least one gene. You can see from this picture, promoter is there that initiates the process of transcription. You can see 5' end. Can you see 5' end? Okay. Then 3' end. Okay. So one side 5', other side 3'. One side 3', other side 5'. Do you follow? So first step, promoter initiates the process of transcription at 5' end. Okay. Next, structural gene codes for RNA and RNA polymerase. Next, terminator. It terminates the transcription at 3' end. That is called as the downstream process. Okay. So this is what is given here. And what is a cistron? It is a functional unit. It is old name for the gene. Polycistronic, what does the word mean? Promoter directs the synthesis of one mRNA. That is seen in the case of prokaryotic gene. Monocistronic means it promotes the direct synthesis of one mRNA that usually translates in only one protein. Okay, so that is what is happening here. Here template strand. It is a DNA strand with the 3' and 5' polarity. Acts as template for transcription and codes for RNA. Coding strand, DNA with 5' to 3' polarity, does not code for any region of RNA during transcription. Next important topic, it is another topic so far.